Peter, Inge. Thank you. Inge is going to talk to us about gene expression.
Good morning, everybody. I would like to first have the opportunity to thank the organizers for the wonderful conference, but also to give me the opportunity to present here my results of the study that we have performed. This study concerns the identification of gene expression profiles to identify immunological responses of cobalt and serum dioxide nanoparticles in two different in vitro lung cell models. It was performed in the framework of the FP6 DIPNA project, and we have performed it mainly at Vito with the collaboration of some people involved also in the project. Well, as most of you already know, of course, nanotechnology applications, they raise a lot of concerns about health effects, and by using consumer products or by being exposed in production processes, we get unintended exposure mainly via inhalation. Most of us, we try to explain all these hazards by applying a tiered approach of characterizing the nanoparticles and then applying in vitro tests and go up in the level of complexity. But we still lack a lot of data on different topics that would like to help us to predict real hazard, such as data on inhalation exposure or the internal dose of the nanoparticles, but also on bioaccumulation, biodistribution. And what we think is also a very major knowledge gap is that we miss specific markers and knowledge of biological mechanisms that could give us tools to predict really hazard throughout these different levels of complexity. So in this study, we used cobalt oxide and cerium dioxide nanoparticles, mainly because both of these are used in automotive catalysts because of their catalytic function, reactivity at the surface. And so these are more prone to be inhaled unintendedly, of course. We aimed then in this study to get more mechanistic insight in their immunological pathways or immunological responses in lung epithelial cells by applying a transcriptomics approach and by identifying molecular markers that were altered after exposure. So for this, we used two different in vitro lung epithelial cell models, which are broadly studied in vitro in the literature already with regards to nanoparticle toxicity. But also they represent two different regions, anatomical regions in the lungs. And what's also interesting in selecting these two models is that they also have different cell culture conditions. So at the one hand, we have the A549 cells, which are mainly immortalized carcinoma cells. They are cultured in medium containing 10% serum, whereas the other cell model are bronchial epithelial cells, which are derived from a normal bronchial epithelial cell and which is transformed with an adenovirus, so they more resemble the primary status. These cells are cultured serum-free in a basal medium which contains growth factor, cytokines, and supplements. So the degree of proteins in this medium is far much lower than in the A549 cells. We used suspensions that were prepared within the DIPNA project and which were provided at a high concentration, a rather high concentration. They were stabilized with the tetramethyl ammonium hydroxide, and they were characterized thoroughly by one of the partners of the project. So we identified the primary size using TEM analysis. The cobalt oxide nanoparticles, they were 7 nanometers in diameter, whereas the serum dioxide nanoparticles were 4 nanometers in diameter. And what we also did is we determined whether there was endotoxin present, which is a major biological contaminant which interferes with immunological studies. So we tested the solutions that were added to the cells, and we didn't find any endotoxin below the level of the limit of detection of the assay. Then to determine the relevant concentration for exposure of our cultures, we prepared, of course, the different solutions. We prepared serial tenfold dilutions in medium, and then applied a neutral red uptake assay for, after 24 hours of cell exposure. And what we saw then is that in the A549 cells, also the highest concentration that we applied was not cytotoxic, meaning that we were able to apply 60 microgram of cobalt oxide nanoparticles to these cells and 12.7 microgram per mil serum dioxide nanoparticles. But when we looked at the B2B cells, which are cultured in harder serum-free medium, we saw that these were much more sensitive to these exposures, as was previously explained by Christopher as well. So these particles probably do not contain a large corona, and therefore we observed that the cobalt oxide nanoparticles were very toxic and went above the 30% cutoff that we took as a cutoff level for deciding on cytotoxicity. Whereas the serum dioxide nanoparticles, they were confirmed by DLS to precipitate in the medium, and therefore they give here the lower absorbance values. So for the B2B cells, we decided to use a tenfold lower concentration. We also verified whether the particles were uptaken by the cells. Unfortunately, we only did that for the B2B cells, because I've learned just now that also the corona is there, a very important trigger to determine this uptake. And what we saw in the B2B cells after 24 hours of exposure is that the cobalt and the serum dioxide particles, as you can see here, were able to enter the cytoplasm of the cells. But they're sticked rather confined here in endosome, so they were not really free, I would say. Then we exposed our cells at different time points, as indicated here, in 3 independent replicates, and we exposed them both to the nanoparticle suspensions and the solvent thereof. The RNA was extracted, and we performed a full genome microarray analysis on them. The resulting cellules were then normalized and filtered for outlying values or values that were below the threshold or the threshold of detection or the noise level. Then we calculated fold changes in gene expression of the nanoparticle exposed cells versus the solvent control. These were 2 color microarrays that we used. And on the resulting mean values, or median values, I should say, of the logarithmically transformed fold changes of 3 independent replicates, we performed the wine sample t-test. And with the criteria that are mentioned here, we extracted a list of differentially expressed genes for the 2 different cell types. On this list, we then also performed pathway analysis and functional analysis. So here the first overall time kinetics of the transcriptional response in the 2 cell models. Well, what we observed immediately was that the B2B cells responded more sensitive as compared to the A549 cells, because a higher number of differentially expressed genes were identified. And also we observed for the serum dioxide nanoparticles that these had a higher response in both cell types. There was no clear kinetics in the A549 cells, but in the B2B cells we saw that the number of altered genes was higher at a more progressed incubation time. When we look more into these data, we see that most of the genes that were differentially expressed were downregulated, indicated here in green, whereas only a minor fraction was upregulated. But for the cobalt oxide nanoparticles, there was a gradually increase in time of the upregulated genes, whereas for the serum dioxide nanoparticles, these levels remained similar over time. On the resulting gene profiles, we performed hierarchical clustering analysis, both on the level of the exposure conditions, so these are mentioned here. We have the different nanoparticles for the different time points and for the different cell types. But the clustering was also performed at the gene level, which is indicated here on the left. The red labels indicate upregulated genes, and the blue are the downregulated genes. And what we saw is that within one cell type, most conditions clustered within one nanoparticle exposure, except for the exposure of B2B cells to the cobalt oxide nanoparticles. These also clustered with all other different exposure conditions. Then we saw also that there was quite a time-related clustering for the cobalt oxide nanoparticles, but not for the serum dioxide particles. That was less consistent. And finally, we also observed that there were two highly distinct clusters at the gene level, which are indicated here with the pink and the yellow. And I will zoom in on these now. So these contained for either the A549 cells on the left side or the B2B cells on the right side quite high upregulated gene levels, and were distinct from the other cell type and the other exposure conditions. These were upregulated in response to the cobalt oxide nanoparticles and contained 11 genes in the case of A549 cells, 20 genes for the B2B cells. And when we performed a gene ontology characterization of these profiles, we concluded that most of the genes in these clusters were involved in metabolic processes, whereas also for the B2B genes, there were also a number of genes involved in trigger to stimulus. But to get more functional insight in the data, we also did a gene ontology analysis on the data to extract all the genes that are related in immune processes. And we saw that only a minor fraction of the differentially expressed genes were involved in immune processes. This fraction was between 1 and 14 over the different time points and the different exposure conditions. But again, we saw also that the B2B cells were most sensitive here after 24 hours to the cobalt oxide nanoparticles, so that you can see from the more extensive list in this table. The different markers were involved in the pathways that are indicated here on the right side. So a number or a relatively large group of genes were involved in not yet assigned pathways, so we could not relate them to each other yet. But another bigger group of genes were related to signaling in the immune system. And what was obvious from these observations was that the number of the immune-related genes is rather low in this list, if you take into account that we started from genome-wide microarrays, and also the levels of gene alteration were quite modest. But there was one marker that appeared all through the different exposure conditions and over time, which was AKT. This gene was decreased mainly in the two cell types and could maybe play a role as a generic marker for the interaction between the lung cells and the nanoparticles. So when we looked up this function of this gene, well, this is a protein kinase, which is a key signaling molecule, which has a role in cell proliferation, cell differentiation. And we find two different publications which also could relate the AKT expression or activation, at least, to particle exposure in allergic airway disease or in even in vitro in the B2B cell line. In this last publication, there was an indication that through the activation of the AKT, there was also an activation of NF-kappa beta, and further on they also observed an increased response for different cytokines related to that activation. So to conclude, I think our study showed that we did not induce or did not observe a clear induction of inflammatory response in these two cell lines after nanoparticle exposure, and that the two different cell lines also give a quite distinct response. There were only a few common players. Well, I think the difference in the response of the two cell models could be brought back to their different origin, so either being more primary or cancerous, either being from the bronchial region of the lungs or the alveolar, but also dependent, of course, on the difference in serum levels in the medium. I think this may be the major factor here. So I guess these results need to be validated by extending this study by more studying more nanoparticles, or to go to more relevant cell models that also comprise professional immune cells. So I think we have seen all through the days in the conference that there are already well-established co-culture models that could be useful for this and could be exposed at the air-liquid interface. But in general, I think such a broad transcriptome approach may give us benefit to find and understand biological mechanisms and to try to extract markers that could predict more reliable the hazard that is involved in nanoparticle exposure. Lastly, I would like to acknowledge all colleagues from the DIPNA consortium that you can see here that worked on this study, as well as my colleagues at VITO that also had a role in achieving this work. Thank you for your attention.
Time for question, Francesco?
Well, I have to disappoint you. We didn't do so far, so we don't have these data.
Thank you. I think we'll move on to the next presentation. Thank you, Inge. Well, for your scene of asking the question, we ask you to come and give a presentation now, Francesco. So